Welcome, folks, to Chapter 6, Microbial Nutrition and Growth. So in Chapter 5, in our previous chapter, we had talked about energy. And we learned in Chapter 5 that energy and nutrients are intimately tied together because oftentimes when we're doing energy transfers, we're actually utilizing the chemical bonds within nutrients to either extract or store or transfer or move around energy. Same for electrons. We're using electrons from the nutrients that organisms, including microorganisms, are taking in to make those energy transfers and to maintain the metabolism that they need to maintain their organized complexity within a universe that's constantly moving towards entropy, right? And so that input of energy via nutrients or transfer of energy via nutrients is really essential. And so these two chapters are kind of parallels. And I would recommend that if you want to review the notes from Chapter 5 or any previous notes, that you go ahead and pause this video, review those notes, and then come back into the video. Okay, so let's start our discussion around microbial nutrition and growth. One thing that you're going to see in this discussion of today's chapter is that it's really kind of an overview of the rest of our lab. We're going to talk about all of the different techniques or most of the different techniques that we're going to be going through for the rest of the lab. And so I definitely want you to take notes on that as well. And so essentially the three parts of this chapter are: one, what are the nutrient and kind of environmental requirements for a microorganism to grow? Okay, and so that can be a diagram where you kind of say, okay, what are the carbon requirements? What are the energy sources? What are the electron sources? What is the oxygen requirement or the environmental oxygen that can be handled by that microorganism? And that you can kind of envision or imagine as one diagram. So that's part one. Part two talks about, okay, now that we know these requirements that were part one, part two of the chapter discusses, okay, well, what are the lab techniques that we use to kind of explore those specific questions or those specific microorganisms. And so we'll go through a series of discussions around different types of media and how we kind of utilize those media to grow and explore various types of microorganisms. And then the third part of this chapter is looking at microbial growth and how that looks in terms of the various phases and stages that microbial growth goes through when we're culturing them in the lab or even in the environment, although the discussion is a little bit different between the lab and the environment. Those three parts of the chapter will be divided up into the two videos that we will have for this lecture. And so my suggestion is from the get-go, just be prepared to generate three different diagrams, right? A diagram of, it could be a concept map, it could be kind of a flow chart, some sort of diagram that specifies the requirements for growth of a microorganism. And you're going to include carbon sources, energy sources, electron sources, and oxygen requirements. And then a second diagram that details out the different types of media and methods that we use in the lab to then explore organisms with those various nutrient requirements. So that's the second diagram. And the third diagram is essentially going to mimic chapters, the 20th figure, so chapter 6, figure 20, where it shows the different phases of growth, and then you can kind of detail out what's going on in each phase. And of course, I welcome you guys to email those diagrams to me, share them with me for feedback. And those will kind of be your guide for studying for this chapter. So let's jump in. Okay. So as we mentioned, microorganisms need to grow and reproduce to maintain their individual homeostasis as well as their population. Okay. And when we use the term growth in microbiology, we're not usually talking about an organism getting larger or kind of growing over time, because oftentimes these are individual cells and they just kind of are the size that they are. What we're talking about usually is population growth if we say growth within a microbiological setting. Now, growth, as we've seen in the lab, can look like colonies on a plate, but also in the environment, oftentimes microorganisms are aggregated together in biofilms. We are also familiar with biofilms in our kind of lives because there are biofilms on the plaque on our teeth, or various types of biofilms that we need to clean out of kitchen, restrooms, off the sides of boats, so on and so forth, that accumulate. So here you see individual colonies in a lab setting, but within the environment, they're not individual colonies. They're actually aggregates or associations of different organisms living together, utilizing various aspects of the same environment. Okay. So when we're looking at microorganisms in the environment, sometimes we need to culture them in the lab. And even just without culturing them in the lab, understanding how they interact with their environment is an essential piece of understanding those microorganisms. Having that information, though, allows us then to culture them in the lab as well as part of that exploration. And so there's some primary kind of characteristics that we need to know about. Carbon is the basis of all living systems, so we need to know what is the source of carbon for this microorganism. How does it utilize, or how does it extract carbon from the environment? Is it just using CO2 that's just in the atmosphere and building up its own carbon molecules, biological macromolecules? Or is it not able to utilize the carbon that's just in this atmospheric source, carbon dioxide, and they are actually having to extract carbon from organic molecules that already exist, essentially eating other organisms that already built up those macromolecules. So carbon is one. Oxygen requirements are another. So oxygen is toxic. Too many organisms, even organisms that utilize it, have to have some sort of antioxidants or some sort of method to actually detoxify oxygen. Nitrogen is a limiting source. It's required if you think back to biological macromolecules, nucleic acids and amino acids. So genetic information and proteins are essentially built off of nitrogen as well as other elements. And it is also in other types of macromolecules as well. And so knowing where the nitrogen source is or how nitrogen is being incorporated sometimes becomes important. And then hydrogen is essential. Why? Who remembers what is kind of essentially tied to hydrogen movement in cells? Okay, I'm hoping that you're thinking back to proton gradients and electrons and the utilization of energy via those two gradients, electrons and protons, to generate ATP or to kind of energize various metabolic reactions in the cell, okay? And so looking at where a microorganism is getting its various requirements from allows us to kind of understand how it's interacting with its environment and with others in its environment. Okay. So we start off with the source of carbon. And so you can think of the source of carbon as two sources, either autotrophic, auto meaning the organism itself, auto, is able to build up the carbon molecules. And so that source is carbon dioxide. Whereas hetero meaning that they need to get those requirements from somewhere else. And so those are organic molecules that they're getting them from. So heterotrophs are having to ingest other organic molecules and bring those in and then actually utilize those. In terms of the source of energy, we have phototrophs that are able to actually use the solar energy from the sun, and chemotrophs, which have some sort of chemical energy that they are going to be using or interacting with. And so we can usually kind of categorize organisms based on these four categories. that are based off of what the source of light of the source of energy is. Is it light or chemicals? And what is the source of carbon? And so photoautotrophs are organisms that able to use the energy from the sun to actually incorporate the carbon from carbon dioxide into a larger macromolecule, whereas chemoheterotrophs are needing some sort of energy source that is inorganic, and they are utilizing carbon from organic molecules that kind of have already been built out by someone else. And so this kind of gives us an idea of where an organism is getting its carbon and energy sources from. Now, also looking at the electron source, so we said that hydrogens and electron sources are really important. You either have organotrophs that are getting their electrons from organic compounds, or lithotrophs which are getting them from inorganic compounds. And so we have to have a discussion about oxygen to have kind of to continue here. So essentially, oxygen, you'll hear foods that kind of they market antioxidants and this and that, and reducing free radicals. And so oxygen is a really, really, really good electron acceptor, and can be a really good kind of essential part of energy transfer in cells. It's very efficient. But the fact that it's so efficient also makes it dangerous for cells, because it can start transferring electrons from atoms and molecules that it's not supposed to transfer them from. And so optimally, an organism does want to be able to use oxygen, but sometimes it can't. And so in that case, it will have to use some sort of other electron acceptor instead. Okay. So we also want to know where the source of electrons, what's the source of electrons, and where are the electrons kind of going? What is the final acceptor of those electrons, right? And so the electrons might be harvested off of organic molecules, so we'd call that an organotroph, and they might be harvested off of inorganic molecules. And the reason for this is essentially comes down to the characteristics of oxygen, the reason for how you harvest electrons and who accepts it. Because essentially, oxygen is one of the best electron acceptors within these kind of metabolic reactions. And that is because it's able to transfer electrons really well. But that same characteristic, that oxygen is such a good electron acceptor, makes it dangerous within the cell, because sometimes it starts moving electrons around that are not supposed to be moved, that are part of a protein enzyme, for example, and are not supposed to be rearranged. And so when oxygen is in a state where it's reactive, we call it a free radical, and any kind of complementary molecule that's able to kind of stabilize that oxygen so that it won't be moving electrons around that it's not supposed to is called an antioxidant. And so that tells us, you know, sometimes in our food, there's marketing around antioxidants and free radicals and this and that. And that's essentially what's going on there. And so optimally, organisms are able to utilize electrons and then donate them to an oxygen, which then becomes a water molecule. But oftentimes there are organisms that just don't have the mechanics to kind of stabilize those free radicals that come off of oxygen use, and therefore they cannot utilize oxygen. Okay, so I'll just show you this quick video here that okay, so this video allows us to kind of see what's going on with that a little bit. So oxygen exists in many different compounds, and it comes into our body in various ways. And it tends to exist originally in some sort of bound form. So the most simple form of oxygen is to have two oxygen atoms that stabilize one another. And we're going to look at what that means. Like, what does that mean to stabilize an oxygen? But there are other ways that oxygen can exist. It can be part of a larger molecule where it is, again, attached to something else and it's stabilized. Well, why does oxygen need to be attached to something else to be stable? Well, you can see that here, that in its atomic form, so just pure oxygen, one oxygen atom, oxygen has what's called a valence shell that is not full. So review of chemistry, right? There are different shells that electrons exist in outside of the nucleus of an atom. And those shells need to be complete to be stable. When you have electron shells that are not complete, specifically the valence shell, which is the outer shell, which is not complete, then that atom tends to be reactive and it tends to be seeking out interactions that allow it to stabilize or complete that shell. And so oxygen tends to be very reactive in its atomic form because it has these two electrons here that are in its valence shell or its outer shell that are not paired. And so you have these electrons that are paired, 1, 2, paired, 3, 4, paired, but 5 and 6 are all alone. And so they're seeking out interactions. And essentially the best way to kind of stabilize this atom is to then find 2 more electrons to pair up with. And that's what water is you have a hydrogen atom that comes in from somewhere else, and it actually interacts with or shares one of these electrons and one of its own electron, and then this other electron and the other electron that's in the hydrogen atom, and then you get H2O, right? But when we start to separate these oxygens and put them through electron transport chains and stuff, essentially that's great because these very reactive electron kind of regions then accept electrons readily, and then that electron transport chain can continue. The problem is when there is oxygen that is unstable, that somehow is not paired directly with an electron transport chain or is not paired directly with the flow of electrons in a metabolic reaction, then, and we'll see here, then instead of accepting the electrons that it's supposed to accept off an electron transport chain, it starts seeking out electrons from other atoms and molecules. This might be a protein that is doing a specific reaction in a cell. This might be a macromolecule that's building up some sort of structure within the cytoskeleton of the cell, right? And if it starts to then steal electrons off of structures that are already built up, that are not meant to be exchanging electrons, that are not meant to be destabilized, then it will cause damage in those structures. And so that's what we see here is this free radical is now exchanging electrons with atoms or molecules that it's not supposed to be exchanging electrons with. And in that way, it is stabilized, but then it makes a chain reaction that just continues to destabilize those structures. Okay, and so that's why when we say, free radicals and antioxidants, that's what we're talking about. And so an antioxidant is essentially a molecule that, based on its molecular structure, it has electrons to donate, right? It's just, the way that its molecular structure is, it can actually donate electrons to the free radical or the oxygen that is kind of seeking out those electrons. It stabilizes the free radical, but it itself does not become unstabilized. The antioxidant itself does not become unstabilized. And these antioxidants are natural within kind of a natural, balanced, diverse diet. And then our body utilizes them to kind of essentially stabilize oxygens that are moving through our cells so that they do not become, or they do not act as free radicals and start to destabilize our cells. And so this discussion is essentially connected to how organisms are utilizing oxygen or existing within the presence of oxygen, right? And so you have organisms that have the mechanisms, the antioxidants, and the various kind of safeguards to protect themselves from potential free radicals. And you have cells or organisms that don't. And so, and then you have cells and organisms in between, right? And so, if an organism is able to use oxygen, and that's the predominant way in which they kind of extract their energy via their nutrients, then we call them obligate aerobes. Aerobes referring to oxygen or air. If you have organisms that are completely unable to utilize, not just utilize oxygen, but even exist in the presence of oxygen, because they don't have antioxidants, they don't have safeguards against free radicals, we call these obligate anaerobes. Anaerobe, so an meaning no, and aerobe meaning oxygen. Okay. And then, of course, there's a spectrum, because in biology, you always have some sort of spectrum and you have some sort of exceptions. And so essentially, the spectrum goes from obligate aerobes that are able to exist within an aerobic environment to anaerobes. And between that, you have facultative anaerobes, which they prefer oxygen, but they can also tolerate some anaerobic conditions. And these are usually cells that can do both cellular respiration and fermentation, just depending on the presence of oxygen. So I think for me personally, I think it would be easier to think of these as facultative aerobes, because they are aerobic, they are able to utilize oxygen, but they can also make an exception and also grow when there's no oxygen as well. So they're called facultative anaerobes, but just in my mind, it makes it easier to say facultative aerobe. And those concepts are essentially interchangeable, but you would want to use the formal term facultative anaerobe. And then you have aerotolerant anaerobes, and these are anaerobic organisms that they don't utilize oxygen, but also they have the machinery, they have the mechanics to protect themselves from free radicals. And so oxygen isn't toxic to them. So they're not using the oxygen, but they're also not bothered by the oxygen. And so that's kind of your spectrum. If I was making a diagram for your study diagrams, I would just make a spectrum of, oxygen tolerance to no oxygen tolerance. So obligate aerobe to obligate anaerobe, and then put these in the middle there. Okay. And that's what we had just discussed here. So we talked about carbon, we talked about oxygen, we talked about hydrogen in relation to electrons. There are other nutrients that are essential to living systems: nitrogen, sulfur, magnesium, calcium, copper, iron. But the most important, or not most important, but most in terms of biomass, if you look at the dry weight of an organism, out of these is nitrogen. So if you take a cell and just dry it of water and then measure the amount of nitrogen that's in a cell, usually it's about 15% that is actually made up by nitrogen, because there's proteins, there's genetic information, all of that is necessary for these living cells. And so nitrogen becomes important also because of chemistry. And so nitrogen exists in our environment, right? Just like I'm touching nitrogen right now, it's all around me. It's like 78% of the atmosphere. But because of its chemical structure, it's very, very difficult to release that nitrogen into a form that's useful. Okay. And so I'll look in the description for a video on nitrogen. Okay. And in that discussion, you'll see how nitrogen is so stable, essentially, in its N2 form, that when you try to break it apart, it's very, very difficult. Organisms need very specific enzymes for that. So it's very interesting. Nitrogen is all around us, but it's essentially inaccessible to organisms except for nitrogen-fixing bacteria. And so we really rely on nitrogen-fixing bacteria within natural systems. Now, this brings us to kind of a macro example. Essentially, in the last, 100 years, we've been able to, as human beings, extract nitrogen in an artificial method. And that's how we get inorganic and artificial fertilizers, right? And so our food systems are built upon the extraction of nitrogen, but now an artificial method. And so it is because of that evolutionary history where we've always had nitrogen as kind of this limiting source that we've relied on nitrogen fixers to extract. And then all of a sudden, within just, about 100 years, now we've started as humans to release nitrogen into the environment in a very quick way via these artificial methods. And it is because of that that you see destabilization of waterways through eutrophication and algal blooms and things like that. It's because environmental systems have evolved to not have a lot of nitrogen in their environment. Same for phosphorus. And then when it's released all of a sudden, or it's available all of a sudden, usually we have mechanisms to opportunistically use those. And there are certain species that are kind of like weedy species that can then take advantage of that and then destabilize whole communities and ecosystems. And so that is a parallel discussion, but I do want to put it out there because it is these micro processes that then become, discussions around, okay, well, how are we going to get our food? How are we going to produce our food? Can we produce it in a small scale because nitrogen is limited? Oh no, now we have methods for extracting nitrogen in an artificial way. Now we can mass produce food. And so we can have these very large monocultures. And then what does that do? Then we have to produce other chemicals to actually maintain those monocultures. So it really changes the way in which we interact with our environment and the way in which we feed ourselves and kind of these macro structures as well. Let me actually show you what a nodule looks like. So essentially, there are plants that create symbiotic relationships with these nitrogen fixers so that the bacteria can grow because they actually need an anaerobic environment. And so the plant will create these little anaerobic compartments on their roots. And then the bacteria will be able to release the nitrogen from the soil. And that's for their own benefit, but then also the plant benefits by getting first dibs on the nitrogen. So here you can see this is a pea plant. Just rewind this. Okay, you can see this is a pea plant right here. And then in the roots, you see these little tumors. Okay, each one of these little compartments here, 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 this one up here, all of those, see right here, here, this one, all of those are little compartments in which the bacteria, the nitrogen-fixing bacteria, are actually living. And the plant creates those compartments, and it's this symbiotic relationship where nitrogen can be released. And you guys are actually cultivating nitrogen fixers in the garden as well, so you get to see some of these organisms in the lab. Okay. So as we said, there are other nutrients that are necessary, like phosphorus, sulfur. There are vitamins, even metals that are used. When we talked about enzymes, we talked a little bit about how you need vitamins or sometimes metals, some sort of cofactor to actually sometimes activate or make an enzyme more efficient. And so those are needed in small amounts. And in a balanced diet, we do usually get the nutrients that we need. Okay. So those are kind of the nutrient requirements for microorganisms. Although that does scale up, other organisms need some of the same kind of requirements. Now let's talk about the physical requirements. So temperature, pH, so on and so forth. When we had our discussion last chapter, we talked about the kind of how essential it was to maintain an environment that is kind of efficient or stable for enzymatic activity, for stability of macromolecules, for example. And so it's the same when we look at the whole organism. Some organisms have metabolic processes that are fit to a higher temperature or a lower temperature or a middle temperature. Same for pH. And that is based off of the environment that they have evolved in over time and the characteristics that they have taken on. And so depending on that organism and which environment it lives in, it will have various characteristics that are most stable for it to maintain its homeostasis, right? And so just using the example of our microbiome, we have microorganisms that live on our skin. Our skin tends to be salty and dry, and so organisms have to be able to tolerate that environment. We have microorganisms that live in our mouth. That's a more moist environment that has a lot of different nutrients that are coming in. And so you have different organisms that are forming biofilms within your mouth, right? Then there's the stomach. The stomach is a very acidic environment, very different from the rest of the body, which is about a neutral pH. And so the microbiota in your stomach are then can tolerate their enzymes, their cell membranes, all of their mechanical kind of metabolic functions can exist within an acidic environment. But they wouldn't be well fitted to a neutral environment. And so depending on where that organism exists, it will have different kind of physical or environmental needs. Anything that lives in and on us exists at about a 37 degrees Celsius when it comes to temperature, because that's our body temperature. But you might have organisms that are in extreme environments that are very high temperatures or very low temperatures, and then they would be well suited to those temperatures. Okay, and so the effects of temperature on microbial growth. I think when we talked about enzymes, we talked about something similar, that generally speaking, as you increase the temperature, molecules and metabolites and things are moving around a little bit more quickly, and that allows you to increase the efficiency of various chemical reactions. But at one point, there's an optimal temperature, and where you have the most kind of metabolic activity without that temperature then destabilizing the protein structure of your enzymes, and then that destabilizing the enzymatic function of those proteins. And so once you get over that, then very quickly the enzymatic activity starts to drop off and the cellular kind of growth and activity starts to drop off. Okay. And so this kind of gives you an idea of the growth rate of various types of organisms. Most of the organisms in our microbiome are mesophiles, which means middle temperature, but you also have organisms that are thermophiles and hyperthermophiles that live in very, very warm environments, and their enzymes and their membranes are fit to those environments. And you do also have organisms that live in very cold environments and thrive there. Okay, so here you're looking at a glacier, and you're actually looking at microbial growth on that glacier. Okay. So that's temperature. How about pH? We talked a little bit about pH already, but pH is similar. In terms of temperature, when you increase the temperature, it increases the kinetic motion of molecules and atoms. And so they're more likely to interact with each other, but they're also more likely to destabilize. Whereas pH is a little bit different. It's more based on the ionic interactions between the solution and the molecules or the biological structures within. And so those ionic interactions then will destabilize the weak bonds that actually allow for proteins to fold, or the membrane to kind of maintain its integrity. And so the pH is really essential to every single system within the cell. And so you have organisms that their systems are generally kind of fit to a neutral pH, which we call neutrophils. But then you do have extreme organisms that are able to live in acidic or alkaline environments as well. Water is also essential to cells. Essentially, the cell is a bag of water, and oftentimes that bag of water lives within water, right? An aqueous solution. And so maintaining that hydration is really important. And so if cells begin to desiccate and actually dry out, oftentimes if they tend to live environments like that, then they'll have mechanisms like endospore production, or they'll have a capsule to protect themselves from the outside, so that they can maintain an aqueous solution within that. And so different types of protections in anatomy to protect themselves from desiccation, which we had talked about when we talked about cellular anatomy as well. Then water is also related to salt content. If you remember back to other classes where you have discussions around osmotic pressure and osmotic tension, whenever you have a saline environment or a solution around a cell that's high in dissolved salts, then that actually influences the water flow of the cell itself. And so the water will actually rush out of the cell to try to dilute the environment. And vice versa is the same. If you have an aqueous solution around a cell that's really low in salt concentration and dissolved salts, then that water will sometimes rush into the cell and it will explode. And so we actually know this anecdotally because we know that if you're stranded in the ocean, you never drink salt water because drinking salt water will actually make you more thirsty. And so organisms that have evolved in highly saline environments, they have to have protections against the movement of water outside of their cell. And organisms that don't have those evolutionary protections, they generally are not successful in highly salty or saline environments. Okay, when we get to our discussion around lab methods, you're going to see that actually some media, some growth media that we use in the lab are salty or have high in salt so that they can actually select out most organisms and just grow a couple of different organisms that are important for medical testing. Okay, so now we have an idea of the different kind of nutrients and environmental factors that we're kind of playing with when we're trying to grow microorganisms or understand how they grow. And so the next part of the lecture will have a discussion around lab techniques to allow us to explore and grow microorganisms using these various ideas. And it will also have discussions around growth of microorganisms within a lab stage and within the environment. But one thing I want to preface that whole discussion with is essentially what we're trying to do. We already kind of peeked at isolated colonies earlier in the lecture. What we're trying to do is explore and understand specific species. And so we tend to isolate them. We tend to talk about them on isolated plates, pure colonies, utilizing different types of metabolites, so on and so forth. But always remember that in the environment, organisms exist within community, okay? And so it's not just one isolated microorganism that's growing on a plate that has just the nutrients that it needs to grow. It's many different microorganisms that are interacting with one another. And so these methods allow us to isolate organisms, allows us to explore them in isolated or in purified cultures. But we also need to make sure that we're understanding the bigger picture and the interactions between these organisms, which will not be our focus in our lecture today in chapter 6, but we will throughout the term kind of go back to those interactions. And so just reminding you of the big picture is really essential here. Okay. What are some of those interactions? Well, you have antagonistic interactions, just like in the human world, for example. You have antagonistic interactions where microorganisms will produce antibiotics to kill off other organisms. We utilize some of those antibiotics in the medical field. You have synergistic relationships where kind of organisms are existing in parallel and kind of, existing within community with each other. And then you have the next step, which is symbiotic, where organisms are actually benefiting one another in their environment. And oftentimes they do this within biofilms. And so I'm just going to show you an image that's the same as that slide there. And biofilms are when organisms, the initial organism, and you can think of this in the macro world as well, right? You have maybe a barren field or something. You have an initial organism that comes in and says, oh, this looks like a good home. And then you start to have an aggregation of different organisms and even different populations around the resources in that same region. So kind of like a little city, but on a micro scale. So you have organisms that come in, they see that there are nutrients in that area that are to be, ready to be utilized. And then they'll actually release different types of macromolecules that help them kind of, adhere to that surface, whatever it is, wherever those nutrients are. And then you'll have other microorganisms that have what's called quorum sensing, and they will actually sense the density of other organisms around them, and they will move towards areas where there's a higher density of microorganisms because that tells them that there's nutrients there. And this is the same in the macro world. So, move out of the micro world for a second with me and imagine that you're in an airplane at night, right? And when you look down, you can see where city centers are, right? And oftentimes people will move into those city centers because that's where there's an aggregate of resources for the human population, right? And so microorganisms are doing the same thing. They're sensing out where is it, back to the micro world, where is it that other microorganisms have found, living to be, possible. And so that must be a place where there's nutrients, where there's not toxins, where there, whatever needs to be utilized is there. And so they will actually aggregate in those areas via quorum sensing and seeking out those microorganisms. Okay. And so when you have that aggregate, that's when you get biofilms, because those organisms say, okay, this is a place to be. In this mouth, there's food coming in all the time, and there's moisture, and there's even enzymes. So the human body produces enzymes that help break up that food. So the microorganisms don't necessarily have to produce them all. So this is a great place to live, and then they'll start to make biofilms. Same with medical equipment, either in or outside of the body. So being aware of those there. In the environment, both kind of in the external environment and even in our homes, we have these biofilms. Okay, so on that note, we're going to go ahead and pause this first part of the video. And then the second part of the lecture will go through lab techniques and then microbial growth. Okay, and so like I said, you can have like 3 diagrams for this class. You should be able to complete that first diagram on what are the nutrient and environmental needs of microorganisms via this first part of the lecture. And then the other 2 diagrams, one on how we use this information to generate lab techniques that allow us to grow microorganisms and explore and understand microorganisms in a lab setting. That will be the first part of the next video, and the last part of the next video will talk about growth of microorganisms within the lab and the different phases that they take to as they grow. Okay, so I'll see you in part 2. Okay, so I'll see you in part 2 of the video.